Bonjour, good morning, everyone. My name is Emmanuel Lebrun-Damiens. I am the French Consul General here in San Francisco, and it's my great pleasure this morning in San Francisco to be with Nevan Krogan, who is the director of the Quantitative Biosciences Institute, and Christophe, who is joining us from Paris. He is the scientific director of the Institut Pasteur. It's wonderful to be with you this morning to talk about your scientific partnership and how you fight against COVID-19. I'll ask a few questions and then we'll have questions from the audience, and I'll relay the question to you. But perhaps first, if I could ask you to tell us how you met. It's been a long-term relationship. It's a French-American collaboration. How long is it, and how did you guys come to this wonderful partnership? Perhaps Nevan first?
Sure. It has been a delightful interaction over the last few years. I've always enjoyed my times going to the Pasteur Institute, and Christophe and I have known each other for many years based on some of the work that we've done. This particular partnership started a couple of years ago. We were at an event in Paris, and we went to visit Christophe. This was before he was the director of the Pasteur Institute. And we were talking about ways in which the Pasteur Institute and QBI could work more effectively together, because we feel our strengths are very synergistic. And we came up with a number of plans, including a symposium series, which did start in 2019. The first one was here in San Francisco in March 2019, and a number of scientists came over from the Pasteur Institute. A number of collaborations were started. We were supposed to have our second one, March 2020, in Paris, and for obvious reasons, we were unable to do that. But the relationships that had been forged over the last few years bore a great deal of fruit in the context of this pandemic, I would argue, because we were in a great shape to start working together, bringing our respective expertise to the table in a very collaborative way to try to come up with solutions for this particular pandemic we're all dealing with.
Thank you, Nevan, and we can see all of you on this picture. Christophe, perhaps your memories of this meeting.
Well, I think it was a very fruitful meeting. We had brought people with very different expertise from Institut Pasteur. So you can see Félix Rey, who is one of our structural biologists. Olivier Schwartz at that time was the scientific director, and he's a virologist, and he's currently, both are actually very much involved in working on SARS-CoV-2 and COVID-19. So it was a very fruitful meeting. Actually, at that time, we decided that we should really create a partnership between QBI and Institut Pasteur to work on different strategies to fight against infectious disease. And we were about to sign a memorandum of understanding between our two institutions in January when COVID-19 started. And so actually we didn't have to sign the memorandum of understanding to start really collaborating actively between our two institutions. And this is actually thanks to Nevan contacting me and proposing to set up a strong collaboration to work on COVID-19 and strategies for developing antivirals. So, yeah.
So let's move to COVID-19 and let's talk science now. So we're working on something which is called therapeutic repositioning. Nevan, could you explain us what it means and tell us how you developed this special collaboration with Institut Pasteur on COVID-19 and on therapeutic repositioning?
Right, great. So we at QBI here started QCRG, the QBI Coronavirus Research Group, involving dozens of different groups here at UCSF. And one of the goals of the QCRG was to look at the host, to try to understand how the virus comes in and hijacks our cells. Which genes and proteins does the virus need in order to infect us? And we used a variety of approaches, including a proteomic approach here to generate a map, which is pictured here. And then the logic was to look at the human proteins that we think the virus needs to infect our cells, and then essentially cross-reference that with a drug and compound database. This was work spearheaded by Kevin Shokat and Brian Shoykat here at QBI, and try to identify drugs or compounds that would target one of the human proteins we think the virus needs. Obviously, these drugs and compounds have been developed for other diseases, but through drug repurposing, the hypothesis would be that, okay, the virus needs these human proteins, and if we have a drug that targets one of these human proteins, maybe it could have antiviral effects. And at the time we had made these predictions, we did not have the virus propagating here at QBI to do the test, although there's been a lot of great work recently, especially from Melanie Ott here at the Gladstone Institute, who we're starting to work with. But at that time, which was about mid-March, we really leveraged the relationships that we built with Christophe and Olivier and Marco Vignuzzi at the Pasteur Institute. And they had the virus working, they had the virus growing, and we sent them a number of drugs and compounds, which they were able to test and identify a number of drugs and compounds that look very potent that we're continuing to study in collaboration with Christophe and his team.
So, Nevan, you sent these drugs and compounds to Paris. Christophe, what did you do with it? What happened next in the next phase?
Yeah, so actually the work that has been performed at Pasteur has been performed in the group of Marco Vignuzzi that you just saw in this short movie. Basically, what this group is doing is cultivating the virus in interaction with cells and evaluating whether some of the compounds that have been sent by QBI are preventing proliferation of the virus. And so a number of the compounds that we received actually show antiviral activity. And what is very important is that they show some of them show very potent activity. And so many of these compounds have already been used or developed. And so they are known to be without side effects. And what is really interesting there is that they can very rapidly enter clinical trials for activity against COVID-19. So that's actually the beauty of the strategy that was launched at QBI, is to focus on drug repurposing so that this would speed up the development and the possibility to treat COVID-19.
How was it to work together? I mean, you are 9 hours time difference apart, 11-hour journey by flight. It's 2 different languages, 2 different countries. How was it to work together? You've known each other for a long time now. Can you tell us a little bit what the daily life was in this partnership?
Well, maybe I could speak first. It's amazing what can be done by Zoom. I think we're all learning that. And obviously there's a big time difference, but we'd get up early. We'd be up at 4 or 5 a.m. Discussing experiments, discussing results with a scientist at the Pasteur Institute. And I have to say, it was remarkable to me how fast everybody worked in Paris, and how open they were to collaboration, and how open they were to sharing all the information they got as soon as they got it. And to me, that's why we were able to move so fast. So a lot of Zooming, a lot of emailing. A lot of the scientists are on Slack now, communicating. So to me, it was quite remarkable how fast we could move. And it's exciting to me because you have scientists working essentially 24/7 when you do this collaboration around the world. So to me, that's a very efficient way of doing science.
And so, Christophe, how did you send results of your experiences from Paris to San Francisco?
Well, so I wasn't directly involved because everything was done in Marco Vignuzzi's lab, but things were communicated by, drugs were shipped by FedEx and results were shipped by email. I think what is striking with this pandemics is that it has totally changed our way to do science, because we used to meet, we used to discuss and in persons about potential collaboration. And in this case, I got a call from Nevan one evening at 6. It was 9 in San Francisco, and immediately we discussed about the possibility to collaborate. He knew that we had isolated the virus. And so then I contacted Marco Vignuzzi, Olivier Schwartz, to know whether they were willing to do the experiments. They decided to go on. And so everything has been done through the web after that. And this is really changing our way of doing research, of collaborating. And I think it's kind of, it's both frightening and exciting.
So let's move to the results now. Nevan, could you tell us a little bit? You've made the headlines of local newspaper, the San Francisco Chronicle, but national newspapers as well. You made the front page of the New York Times. So what did this partnership bring in terms of science looking for a treatment for COVID-19 at the day of today?
Well, one of the end goals, obviously, is to come up with a therapeutic or a drug or a cocktail of drugs. And using this pipeline that we put in place, we've now been able to narrow down to 12, 15 drugs and compounds that look incredibly promising. And as Christophe alluded to, several of these are actually now in clinical trials, not being led by us, but companies have been looking at our data and have been going forward with some of our results. We're now actively trying to get some of our drugs into clinical trials ourselves, but the end result of this is that we've narrowed in on a number of drugs and compounds that we're very excited about, that hopefully can contribute in some way to coming up with a treatment for COVID-19. And I truly believe it will be a kind of a combination approach that will be brought to bear, similar to what was done with HIV. There's a cocktail of drugs, and maybe one of ours is one that can be contributed to the cocktail of drugs or biologics. But in the interim, through this collaboration, we've actually found out a lot about the underlying biology behind infection. And a lot of the data that we've generated collaboratively has obviously been disseminated around the world, and that's helping other people to further understand the virus and come up with novel therapeutic strategies as well. So we're hopeful that if we don't contribute to coming up with a therapeutic, that at least directly, at least indirectly, through a lot of the data that we've been generating, both at QBI and the Pasteur Institute.
Is this a strategy that we could use for other illnesses?
So,
Nevan?
So I think, so this idea of drug repurposing has been, used in a number of pathologies and is proving successful. I can give the same example of our institute in Korea, in Seoul, and they have done a drug repurposing screen to see whether some drugs could actually impact on the replication of SARS-CoV-2. But this was, so the approach that was developed at QBI is different, because it was actually based on characterizing a protein map and making hypotheses about drugs that could actually act on the replication of the virus. So I think the approach that's developed at QBI can be extended to many other viruses. I know that Marco and Nevan have discussed about other viruses that could be targeted by this approach, and maybe Nevan can give us more about that. So clearly this is an avenue for discovering new therapeutics. One should not forget about other approaches like systematic drug screening, drug design, but this is one interesting way of finding new drugs and going fast, and this is important in the case of the COVID-19 pandemics.
Maybe I can add to Christophe.
Nevan, perhaps? Yeah, please.
Just to kind of reiterate what Christophe is saying about this is one approach. There's other ones that are hopefully prove fruitful in terms of developing vaccines, developing drugs ultimately from scratch developing biologics, nanobody, antibody type of treatments. So we have a number of tools in the toolbox here that hopefully at least one of them will prove fruitful in the near future. And this idea about looking across different diseases, at QBI, one of our mantras here is that we've developed technologies that is disease agnostic. And we're applying it to many different pathogens, which is why it's such a great relationship with the Pasteur Institute, but also other diseases such as cancer, neurodegenerative disease, heart disease. And when you take a few steps back, you see the same genes and protein come up again and again across these different disease areas. It's the same genes being mutated in cancer that are being hijacked by HIV or coronavirus. And so to me, it's not such a big surprise that there could be an anti-cancer drug that could have antiviral effects or vice versa. So at QBI, we're really about making connections between different labs, between different institutions, and between different scientists working on different disease areas. And I believe there's big discoveries that can be had when you bring together scientists working on the different disease areas, because there's more commonality than people realize.
Thanks a lot. I'll move to a few questions about the future, but before that, I'd like to remind everyone in the audience that you can ask questions to our guests, Christophe D'Enfer and Nevan Krogan. Don't hesitate, I will relay your questions to our guests today. So if we move to the perspectives, my first question would be, what are the next steps in your research? What's going to happen now in the coming weeks and months? Who wants to start? Christophe, perhaps.
Well, so I think what is ongoing in Marco Vignuzzi's lab at the moment is further evaluation of some compounds. That's number one. So the first work actually highlighted a number of interesting compounds, but now you can use medicinal chemistry to expand the range around an interesting molecule, and you need to evaluate whether these diverse molecules are also effective and maybe have better effectiveness than the ones that were first identified. So this is something that is ongoing. The other thing that's very important is that the approach that was developed by QBI highlighted a number of cellular proteins that are important for viral replication. And what the group of Marco is doing at the moment is testing using gene inactivation approaches, whether these proteins are really important for viral replication. And so this will give us targets for drug development besides the drugs that have already been identified. So there are a number of directions that can be taken. We have also a number of now animal models to test COVID-19, and so the compounds could also be evaluated in animal models. So there are, it's opening a number of ways, and I think that's exciting.
Nevan?
On our end, going deep more mechanistically, more biochemically, biophysically, structurally on a number of the, looking at the number of the viral proteins and the interacting host partners. A lot of great work here being led by Klim Verba, Oren Rosenberg, James Frazier on that front. Going deeper on the chemical biology front with Kayvon Shokat and Brian Shoykat leading the way. And then, as Christophe's alluded to, genetics. Genetic perturbation approaches like RNAi or CRISPR, removing genes one at a time or in combination and seeing what effects they have on viral replication. And we've been working closely with the Pasteur Institute and Marco Vignuzzi on those analyses as well. So it's really about combining different approaches, proteomics, genetics, structural biology, chemical biology, to get a better understanding to point us in the right therapeutic direction.
So I have a question from someone in the audience here, which really is in tune with what we're saying here. Is there something special about how the French or Americans do science that has strengthened this unique collaboration? Who would like to take on this question? Nevan?
Okay, I guess what I'd say in this particular case, the first caveat I'd say I'm Canadian, so I should just put that out there. But in this particular situation, I think it's the strengths of the respective institutions that are so synergistic. You have the Pasteur Institute, which is just world-class in terms of studying infectious disease, not just viruses, but bacteria and parasites and fungus. And then at QBI, a lot of labs here are focused on developing technology, disease-agnostic technology that can be applied to pretty much any disease area. And we've been doing a lot of work on infectious disease. So for me, it's a perfect marriage between the scientific goals of these two institutions, and we're bringing both our strengths to the table and having just unbelievable and fantastic results.
Christophe, on the same question.
So there is a second caveat, that is, I worked 2 years in San Francisco, and so to have a collaboration with people in San Francisco for me is really a pleasure. And I think also Pasteur is very international. So, Marco Vignuzzi, that doesn't sound very French. And his team is composed of people that come from France, but from many other countries. And so I think it's an international collaboration. And so a number of our people have been trained in other countries. And so this way of doing international collaboration is immediately efficient. So I think we understand each other very, very well, and this is why things are going so smoothly in this collaboration.
So building on that, how do you envision the future of the relations between QBI and Institut Pasteur?
Well, we're, as I said, we did start with a symposium. We're supposed to have our second one in a few months ago. We're going to have another one virtually in the fall. And it is very efficient to have these symposium through Zoom, although I can't wait to get back to Paris and have, foie gras and croissants and wine. But in the interim, we're going to be continuing building these relationships virtually through symposium. Here's a picture of the poster of our first symposium that was here in San Francisco. So our next one is going to be in the fall. Virtually, we'll pretend it's in Paris, although it'll be a virtual symposium, and just to continue to build on the relationships that we built until we can all get together again in person, hopefully in the near future.
Christophe?
Yeah, so our plan was to actually have a formal partnership between QBI and Pasteur, and to launch projects between our two institutions. I think this is still on the agenda. At the moment, we have this very emblematic collaboration on COVID-19, but there are many other diseases where we can collaborate, be it viral disease, but also parasitic disease, bacterial disease, or fungal disease. So I think the collaboration is going to expand. Our collaboration on COVID-19 is one enlightening collaboration, but I'm very optimistic about new collaborations, and we have the evidence that we can collaborate efficiently even without meeting. So that's really good news.
Is it only a bilateral partnership, or do you bring other institutions in the different collaborations you have? I understand, for example, with COVID-19, you have Mount Sinai in New York that brings as well its expertise to your partnership. Could you just explain us a little bit how that works?
Sure. At QBI, we have, over the last several years, been developing international relationships with a number of different institutions around the world, which in this pandemic did definitely bore fruit. So as you say, we've been working closely with Mount Sinai Hospital in New York. We have, during this COVID-19 pandemic, been working closely with scientists in England, in Germany, starting to, collaborations with scientists in China. And then if you look at the United States, in Seattle and San Diego, North Carolina. So I think what we've started here with the Pasteur Institute and QBI is very exciting, and other people are getting involved. And the more people that get involved, the faster we can move. And this is exactly what we've seen during this pandemic.
And building on your experience, perhaps I'll start with Christophe, but what could help bring more French and American scientists work together the way you did to make scientists perhaps in other fields benefit from international partnerships like yours? What did you learn in your experience that you could use as recommendations for other partners? Christophe?
So I think we need to know each other. When I was a PhD student, I was pushed by my supervisor to do a postdoc in the States, and I know people there, and I'm this is pushing me to develop collaboration with colleagues in the States. I think science is really collaborative at the moment. We see that in Europe, and we have a number of collaborative projects that bring together people from Germany, Netherlands, and Spain, etc. And we need to also have a joint project between the US and Europe. And I think NIH is promoting this. I think Europe should promote that as well, because this is really helping our collaborations. And we cannot just collaborate between European countries. We also need to collaborate with our US colleagues. Obviously, to be able to come back to the States will help us collaborate better. But at the moment, it's a little bit limited.
I'm glad you bring the European Union in the discussion. Each member state of the European Union invests in research, but the European Union as a whole is also a huge investor in scientific research. And the programs that are developed are massive, including on health. And I'm glad you raised that. Before, Nevan, I ask you the same question. Hélène from the audience was asking a question which is very close to mine. She was asking what can be done to ensure this type of dynamic collaboration and very efficient inter-team work will be applied to other projects in the future. Would you like to reflect on that?
Sure, and it is definitely related to the discussion here that's been ongoing. Kind of building on what Christophe was saying, you need to build up trust between groups. You can't just snap your fingers and bring all these scientists together. You got to bring scientists together and get them to interact, get them to build up the trust. And I think the activities that we were doing with the Pasteur Institute are exactly the type of activities that should be done, and more of it should be done, bringing scientists together from different areas of the world, different ideas, different disciplines. So continuing on along those lines. But of course, how do you make this happen? Well, it's funding. And I think that we need to put more pressure on our respective funding agencies to not only put more funding into collaborative research. There's a certain push here at NIH to go away from that. I couldn't disagree more from that approach, is to put money into collaborations, and also to put money into collaborations between scientists around the world, not just for NIH, not just here in the United States. And Christophe and I are been talking how to creatively raise money to facilitate these very exciting collaborations in maybe more unorthodox ways. So with a lot of the attention that we're getting, I hope that we can be successful along those lines as well.
So that brings me to my last question, which was, how can we help you? So you mentioned funding and fundraising, of course, but in terms of assistance that people who are perhaps in the audience or other organizations could bring, what are you looking for? Who would like to start? Perhaps Christophe in Paris?
Well, I think it's the hardest question you're asking. I think, it in parts comes to funding and how, science can be supported. I think it's important that, the general population supports science, because that's an important thing. In this COVID-19 pandemics, at Institut Pasteur, we are a private foundation. We've benefited from public generosity tremendously, and this has permitted us to do a lot of the research we do on, looking for antivirals, but also developing diagnostics, developing vaccines. So support from the general public is important, also because it's putting pressure on our governments to support science. And I think that's what we need.
Nevan, on this question before we wrap up?
Yeah, I think we got to think short term and long term. I mean, short term, obviously, support from the general public through, philanthropic means would be fantastic. But I really do think we need to put pressure on the higher institutions to reconfigure where funding goes, how groups get funded, and collaboration should be more rewarded. Younger scientists should be more rewarded. So I think we're showcasing that here in this particular pandemic. And now the challenge is to take this spirit, change up the infrastructure of how funding is distributed, and hopefully we can be more productive and have more fun in the future like we're doing right now.
Well, thank you very much for this enlightening and optimistic discussion. We see through this discussion all the benefits from an international collaboration, as you both said, how you brought things in the partnership that perhaps two labs from the same country would not have had the benefit of. So thank you very much, Christophe Denfer, Nevan Krogan, from this discussion today. Thank you to our audience, and good luck to you in your partnership. And we wish this collaboration between QBI and Institut Pasteur all the best. Thank you. Merci à tous.
Thank you.
Au revoir.
Au revoir.